Biochemistry is the study of chemical processes within and relating to living organisms. It combines biology and chemistry to understand how molecules interact and contribute to the functioning of cells, tissues, and entire organisms. By studying biochemistry, we can gain a deeper understanding of how life processes work at a molecular level. This knowledge is essential for developing treatments for diseases, improving agricultural practices, and advancing biotechnology. Carbohydrates. Carbohydrates are essential organic molecules made up of carbon, hydrogen, and oxygen atoms. They serve as a primary source of energy for living organisms. Carbohydrates can be categorized into simple sugars, monosaccharides and disaccharides, and complex carbohydrates, polysaccharides. So these are the monosaccharides, glucose, fructose, and galactose. Galactose is an isomer of glucose, which means it has the same molecular formula, but the structural formula is different. Monosaccharides. Monosaccharides are the simplest form of carbohydrates and are referred to as single sugars. Glucose, fructose, and galactose are the most common ones. There are a lot more, like 20 something, but it's mostly just glucose, fructose, and galactose. And they are quickly absorbed by the body and provide immediate energy. So here we can see the structural formula of glucose, galactose, and fructose. They all have 6 carbons and the same amount. They have the same amount of carbons, hydrogens, and oxygens, but as you can see, it's a bit different where some of the structures are. Like there's an OH group on the outside of galactose here, but it's on the inside of glucose here. And fructose, the carboxyl and the carboxyl and the hydroxyl groups are in different places. So, and the disaccharides, which should be a combination of 2 monosaccharides. So sucrose is a combination of glucose and fructose. Lactose is a combination of galactose and glucose. Maltose is a combination of 2 glucose molecules. The disaccharides consist of 2 monosaccharides joined together, and as I said before, and the digestive enzymes break down disaccharides into monosaccharides for absorption. Polysaccharides. The polysaccharides are complex carbohydrates made up of multiple monosaccharide units linked together. Starch, which is found in plants, well, it's a form of starch found in plants, store glucose as starch. It's a chain of glucose units and serves as a long-term energy storage form. In plants, starch is stored as amylose, which has a branch structure and is made up of glucose bonded with 1,4 glycosidic bonds, and glycogen, which is starch in animals. We store our glucose as glycogen, which is made up of amylopectin, which has also a branch structure, but it's more branch than amylose, and it has 1,6 and 1,4 glycosidic bonds. Cellulose, found in plant cell walls, it forms a tough structure and humans can't digest it, but it's important for dietary fiber. So it would be on the outside of plants like the roughage, so maybe an apple. The outside of the skin of the apple would be cellulose and full of fiber, and it bonds like this, which is a bit different. This is a 1,4 glycosidic bond, and it has, when the 2 glucose, it's beta glucose, when the 2 beta glucose bond together, one has to flip because it can't form the bond normally, which is why it bonds with a bond looking like this. Proteins. Proteins are complex and diverse molecules that play a crucial role in the structure and function of many living organisms. They are made up of smaller units called amino acids, which are connected like building blocks. This is a amino acid. It's made up of an amino group, which is the NH2 group, and a carboxyl group, which is the COOH group, and the side chain, which could be any variation on a hydrocarbon tail. The amino acids are the basic units of proteins. There are 20 different types of amino acid. Each amino acid has a unique side chain that gives it specific properties. Peptide bonds are what link the amino acids together, forming long chains called polypeptides. A polypeptide chain folds and twists into a specific shape, which determines its function. Protein has three structure levels. Primary structure, which is the sequence of amino acids in a polypeptide chain. The secondary structure, which are regular patterns like alpha helixes and beta sheets formed by hydrogen bonding. The tertiary structure, which is the 3D shape, which one tertiary structure that we're familiar with would be collagen, which is found in our hair and our nails. It's one of the strongest structures of protein and most regular one. They have disulfide and ionic and hydrophobic bonds, which help it hold together, which is why they're so strong. And the quaternary structure, which one will be familiar with, is hemoglobin, which is found in blood. It's 2 alpha helixes and 2 beta sheets, all of them bonded together to form the quaternary structure. And the tertiary structure is more common. It's referred to as globular proteins, and it's also what our enzymes are. They're tertiary. And the only way to break them is through heat or to disrupt the bonds. So that would be with something extremely acidic or basic, or something that would ionize it. So it wouldn't be broken by any physical means. Lipids. The types of lipids. You have triglycerides, which are made up of glycerol and fatty acids. They store energy and provide insulation. Phospholipids are crucial components of cell membranes, forming a double membrane layer that separates the inside from the outside of the cells. Steroids, like cholesterol and hormones, like estrogen and testosterone, they have various roles in the body. Fatty acids are the building blocks of lipids. Saturated fats have no double bonds between their carbon atoms and are typically solid at room temperature, like petroleum jelly would be a saturated fat. And unsaturated fat would have one or more double bonds, making them liquid at room temperature. That would be like diesel or any gas that you would use at the gas station. And phospholipids make up the cell membrane bilipid layer. The hydrophobic tails of the phospholipid face inward while the hydrophilic heads face outward. Because the heads are hydrophilic, it means they would rather be in contact with water, so they would face outwards, and in order for the tails to basically repel water and not have to touch it, it would face inwards where there's no water. Nucleic acids. There are two main types of nucleic acids: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA is ribonucleic acid with one less oxygen, hence the name deoxyribonucleic acid. DNA stores genetic information that determines our traits and characteristics. RNA helps to read and use the information stored in DNA. Nucleic acids are made up of smaller units called nucleotides. Each nucleotide has 3 parts: the pentose sugar, a phosphate group, and a nitrogenous base. There are 4 types of nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. In RNA, thymine is replaced by uracil. The sequence of these bases holds the genetic code. DNA has a double helix structure. The 2 strands twist around each other, connected by hydrogen bonds between the nitrogenous bases. The base pairing rules ensure that adenine always pairs with thymine or uracil in RNA, and cytosine always pairs with guanine. There are different types of RNA, each with specific roles. Messenger RNA, it carries the genetic information from DNA to the ribosomes, which is where you'd begin the process of protein synthesis. Transfer RNA helps translate the mRNA into proteins. And the ribosomal RNA makes up ribosomes, the cellular machinery for protein synthesis. The DNA contains instructions for making proteins, but it can't leave the nucleus, so the RNA acts as a messenger. The messenger RNA, during DNA replication, messenger RNA is made and it carries the information in order to make the proteins out of the nucleus to the ribosomes. This is translation. No, this is transcription. This process involves transcription, making mRNA from DNA, and translation using mRNA to rebuild proteins. Changes in the DNA sequence are called mutations. Mutations can be harmful, beneficial, or have no effect, and they are a source of genetic diversity. So right here is a ribonucleic acid. You can see the pentose sugar here, and this is the deoxyribonucleic acid. As you can see, there's one less oxygen right here. The bond that is formed to join together 2 nucleotides is a phosphodiester bond, because the phosphate group on the nucleotide, it has to form a bond with the phosphate group on the other nucleotide. Given that it's a phosphodiester bond, it's also an ester bond, which means it's also a condensation reaction, which is where you lose water from the reaction. And this is DNA replication. You would have the DNA has to form a helix in the 3 to 5 direction, or else it doesn't do it continuously. So for the leading strand, which is this strand on top, it would have a normal replication happening, because when it replicates it would form the 5 strand. But for the lagging strand, should be this strand, it would lose from the 5 instead of the 3, and because of that, it can't replicate it continuously, or the DNA, it wouldn't work well. So they have to use Okazaki fragments and basically put one in and then replicate that one and then take it out and put another one in. So it takes a little longer. And this is protein synthesis. This is tRNA, which has the codon to code for whichever amino acid needs to be made. And once it codes for it, once it has the complementary base pairs here on the mRNA, it releases the amino acid, and then the peptide bond begins to form, and then it goes out again to collect information for another amino acid, and then comes back, and then they repeat that process until whenever. That's it. There's these quizzes. I sent the link for the quizzes in the group chat. Crystal 1, congratulations.